Hello, dear participant. So, did you practice the session for the WGCNA non-trade based data? And if there is any doubt, please do let me know. Okay, so we shall be seeing the inclusion of the trade data now, the second scenario, once the non-trade data has been completed. So we shall be resuming from where we left yesterday, and I will be sharing the code also today. So what is we'll be doing? We'll just be preparing just the data which was included in WGC input. If you have noticed, that WGC input was having in the columns all the gene names and the sample IDs in the row names. This was given as an input to the WGCNA and as well as to the pig gene also. So I am storing this file in dat.expr0. Just I am making a copy of this and I'm transposing it first. So I'm storing the gene names in the row names and again the same format, the sample IDs in the column names. I am just now fetching the array names and gene names individually. So I'm using the array name as an object variable and gene name as an object variable. So gene name will be having all the gene names and array name will be having all the sample IDs from this. So just I'm checking for any outlier or just I'm checking the mean expression across all the samples is uniform or not. So this is the 15 samples and the mean expression is uniform as you can see. And I'm checking for any missing array if there is any. So it is saying that we are actually creating an object called keep array and number my missing array. If any number of the total content in the sample ID or the based on the expression values is less than 500, it will be showing you in the keep array as false. So I'm just creating the array. And in the bar plot, I have created a bar plot for this. And y is actually the, if you can see, there is an inclusion of an additional trade data y is equal to sample info. And we had the age information earlier as we stored yesterday. So this is the age information. So I am storing it in the y and I will be just plotting one outlier dendrogram with this y as a age additional parameter and cluster tree sample. I am just creating the same cluster dendrogram tree which I created yesterday to check for any sample outliers, but just the inclusion of in the x-axis you can see the y variable in that. So there are two different colors, pinkish and you can see the blackish depending on the age numerical distribution which it has distributed for all the samples. So next I have to just create one additional parameter, which is the centrality measure in case of the trade-based data, which is GS. So GS is actually the average correlation value, which we are having the correlation with respect to Y and data. So this we are storing in GS, which stands for gene significance. And the scale free topology and other things are same. This is additional parameter, which I'm showing you how to create that. So I will next be creating one additional dendrogram called the module eigengene network. So this is made in case of the non-trade based data also. So this is all the modules which we got in the earlier case of the non-trade based data. So this is the module eigengene or the representative gene. Module eigengene is basically the representative gene in any of the modules. So ME royal blue, ME green, you can see for all the modules here. So I will just create one additional correlation between the all 20 because there were 20 modules, but 21st, 21 is the age. So I will just eliminating that and creating an internal correlation between all the modules first. And just restoring the sample information and column information from here. So once I have this information with me, so as you can see, this is the module significance dendrogram. And as you can see, the p-value is 0 and significant. So I can get the full module significance value by checking the module significance values. This is the average gene significance values, and then this needs to be taken into account for taking into consideration the hub gene. As you can see, the highest value from all you can see is 0.464, which is of the tan module. Others you can see is less than this. The second least, or the second least one is for the green yellow, which is 0.41. Likewise, you can see in the decreasing order for all the modules like gray 60 is having also zero point 0.4, but the highest one is for the tan. So we can create an additional data with this, and we'll be storing this in an Excel file to create the data, which will be needed for our analysis in case of the trade data as well. So I have just created the GS highest value is tan, which is 0.464. So one trade data which we created using the additional parameter in yesterday, as you saw, which was for the correlation between module membership and gain, which is intra-module connectivity. It will be the same as it is. So for the module membership and the intra-module connectivity, I will be just taking into the same values like 0.18 and just again rerunning all this analysis by changing the module colors here. If you notice that first is cyan and we are having a good correlation value of 3.9 into 10 to the power minus 62, which is significant. So next is suppose blue. I will be just storing this all information in an additional Excel sheet. So this is the same sheet. If you remember, we created with the non-trade based data. So if I just rearrange it again, so based on the values, we can again see the red and blue are having the same highest values in case of the non-trade data as well as the trade data. But the additional two parameters which needs to be taken into consideration is first is, let's take the correlation between the module membership and gene significance first. So I will be creating the values for all the modules again and again. So suppose as you can see for the light green module, the p-value is significant. It is less than 0.05 and correlation is 0.39. Like suppose if we'll note this module again in the Excel sheet and we will again use for another module, which is blue. So for that, it's also significant. Correlation is less than the previous one, but it's significant. So we'll be using this also in the Excel sheet. Just we'll be keeping the data for our analysis. And for the brown one, it's also significant, but the correlation value is less. So this is the data for the gene significance versus the module membership. So this is the data as you can see. And ones which are actually, as you can see, the tan and royal blue, black, purple, gray, 60, light yellow, all these which I have highlighted in yellow, these are actually the insignificant or the non-significant ones as they were having the p-value greater than 0.05. Suppose if you type black here and compute the correlation value of this, so you will be having a p-value of 0.061, which is higher than 0.05. So that means it's a non-significant or insignificant module. So it doesn't matter what the correlation value is this, because we'll discard this anyways based on the insignificance of the p-values. So the correlation values will not matter here. So I have just marked the ones which are in yellow, which is the highlighted ones in yellow. So these are the insignificant ones. And the ones which are in the whitish are actually the ones which are having the significant correlation values. And if we just rank the modules based on the highest correlation values, so I get highest as midnight blue, which is 0.43. Second one is light green. Third is red. So this is the top three ranked orders. Suppose if we rank the modules based on the on the first centrality measure correlation plots, which is GS versus MM. So the third one that remains now is GS versus Kane. So that will be the icebreaker for choosing the a module in our case. So again, we'll be again rerunning this whole chunk of code. Like this code was for the GS versus MM, and the third one we already computed the one which is MM versus Kane. Now this is GS versus Kane. So we'll be suppose computing the GS versus Kane for blue module. It's significant. And likewise, I have created the Excel sheet by changing the module colors again and again in the bottom, which dot color command here. And if we rerun this analysis, suppose for the brown module, it's uh, it's it's very near to the significance level, but still it's significance because it's less than 0.05. So as you can see, I have prepared the GS versus Kane sheet for in the Excel for all the modules, for all the 20 modules here. And the ones which are highlighted in yellow are actually the ones which are insignificant based on the p-value greater than 0.05. And you can list the p-values here in an additional column if the one wants. And if we rank the orders or the modules in the highest values, so first one in this case comes out to be light green, which is 0.47, and the second rank it's a tie between midnight blue and red. So if you notice that light green is highest in this, and it was actually the second highest in this GS versus MM. So in any case, in Kane versus MM, it was anyways the significant one. It was coming out to be the light green one was in the significant zone of 0.99. So in the Kane versus MM, it's also significant. Here it's the first ranker, and here it's also in the first two because it's in the second rank. So we can anyways take the light green as the hub module in our case because it's satisfying the all the three topological measures and their correlation properties from all the modules. So what I will be doing, like the condition which is used for choosing the hub modules. First, we have created the hub modules. Now we have to choose the hub genes. Now this is the trait-based data. So we have to choose a specific amount of genes. So the condition or the default parameter in WGSN is that you have to take the absolute value of gene significance less than 0.2 and absolute value of module membership greater than zero point 0.8 in case of the hub module. So our hub module was light green. So we are accessing the using the dollar command mm.lightgreen, which is module membership dot light green. And after running this command, you will see a number of specific number of genes, like 610 genes out of the total in the light green module. Like suppose if we just count how many are in the light green module using the table command. So in the light green, and it's actually in the all modules, you can see the comparative number of modules is very high because we created the module ligand gene dendrogram at a later point also. So we could see a total number of 610 genes are there, and we will save this file in the hubtrait.txt and just opening that in the Excel. So this is the final file of the 610 modules, 600 genes. This is the hub genes in the our case, and we'll be just copying them and just using the Venn diagram. You have to access the Venn. This is a software used for the Venn diagram and overlapping and intersection and union analysis. So Venn 2.1 is there. In the list one, you have to copy and paste this. So this is the trade data module. You have to type trade data. And yesterday we had the non-trade data also. So this was the non-trade data, and I will be renaming in another box like non-trade data. And we save the hub modules in the case of non-trade data as well. So these were just roughly around double of the ones which are getting in the trade data. So the common you are getting here are 88 in the trade and the non-trade data. So what are actually these 88? You can just click on this box and the result will be shown in the results section at the extreme bottom left. So this is the 88 genes. So I will be just copying them and saving them in the Excel file. And this is the actually, you can just name it as the common, or you can see save it as overlapping or the common DEGs or the hub DEGs, because these are the common hub DEGs between, you can save it as hubs also, between the trait and non-trait data. So trait and T and NT. So if you have any doubt till this session, please let me know and we will be moving with the next session after this. Now we will be representing or recreating the concept of central dogma, which I showed in the presentation slides, by fetching the miRNAs and transcription factors related to these hub hub genes. So one by one, we'll be just fetching the data for our analysis. And what we have to do, we have to just open this 88 genes, which were the common ones in all the hub trait and hub non-trait data, basically modes of WGC and all these. So you have to open Meerwalk first. Meerwalk is a database and it's a highly validated database used for fetching the miRNA and mRNA interactions between the regulatory elements. So after opening the home page, you have to click on the genes option, because first we have to create a edge list, which is the miRNA gene, or you can say miRNA mRNA. So we have the mRNA here, which is the gene here. You have to just copy all this. These are the 88. And after copying them, and once you select the genes here, because we are entering genes, so just copy this and click on submit and proceed. After that, you have to click on the export CSV at the bottom. Okay, so it will be downloaded in your folder. We can just create an additional folder also. So I'm just creating an additional folder, which is FFL 14 September. So I will be renaming this file as, first let me save this file. So this is the interaction between the this is saying miRNA targets. So this is the miRNA mRNA. Now you have to open this file first. And in the Excel, after opening, I am just centralizing all the elements and creating a bold for the headers. After that, you have to go into the binding gap energy and use the data to sort in the increasing to decreasing order. And you will be deleting all the binding gaps energy which are having values less than 1. Like all these are having 0.95, 96, and 98. So I've just sorted in the increasing to decreasing order and I will be just deleting them. After that, you have to check like the position must be only the 3 prime UTR region, not on the 5 UTR region. So as you can see, all are on the 3 UTR region only. You can arrange by decreasing in the increasing decreasing order, and also in the increasing decreasing order. So once you're done with that, you have to fetch the miRNA and the gene column from these two, because it's the first and third. So first we'll be fetching and saving it in the additional Excel file, right? We'll be opening a new Excel file, remove the column which is in between, which is saying refsec, as we don't need that. And this is the file, or this is the H column which we need. Just rename it, because first will be miRNA. We are just renaming it miRNA, and you can name it as miRNA mRNA, right? So this is the first, and just close the Meerwalk file, and I'm just renaming the sheet also for our convenience. Like this is the miRNA mRNA. Now we have to fetch the second edge list, which is the mRNA TF, right? So we again need that 88 genes, which we saved by using the overlapping between trait and non-trait data. So I will be again copy pasting these genes. Now we need the TF and the gene list, edge list, TF and gene edge list. We have completed one edge list, which is miRNA gene. Now for the TF and gene list, we will go to the chia3 database. You have to type chia3 database. And after that, you can see the chia3 database hosted by Icahn School of Medicine. And there will be a box. You can submit your 88 genes here and click on submit. After submitting, you will be seeing two options, like in the you can see option like select a library, and there is a mean rank option given there. You have to just select top rank instead of the mean rank. Once you select top rank, you can see the integrated scale rank here. This is nothing but the p-value. So we'll be taking only the genes which are having the significant p-values. First, let's download this result by clicking on the bottom, download all integrated top rank results at TSV. And after downloading it, I will be just storing or saving this file in the folder where we are doing the exercises. So this is the mRNA TF. We gave the mRNA's input, and TF were returned after that. So I will be now opening this file in the Excel. So after creating the headers as bold, I will be using the cutoff in the score section. Let me just use the data and sort in the increasing to decreasing order, the ascending order. And I will be choosing my cutoff as, for the p-value, we can use the most stringent cutoff also. So the most stringent cutoff I will be using is p-value less than 0.001. So I will be fetching all those TFs which are having a p-value less than 0.001. So as you can see, after sorting, these only three TFs are there which are having p-values less than 0.001, right? Otherwise, they are having at least 0.001 or greater than that. So I will copy these three TFs, right? And saving this in the Excel file we are making for the master list here. Delete this score and library additional columns, as we don't need. We only need two columns. Like here we need miRNA, mRNA. Now we'll be making TF and mRNA. Okay. So we'll be just repeating how many times the number of genes is coming, because in each cell there must be only one element, like one TF, one TF, one TF. Now there are many genes here, so we need to just repeat for that many number of genes, how many for which the TF for it they are occurring, right? So we'll be just copying this TF name again and again, and I will be just segregating this gene list to each of the cell. So for the first TF, I have segregated all the gene names basically, for how many times they were occurring. Now for the second TF, I'll be doing again the same steps. So after completing the manual curation of the TF and repeating number of their genes in the second column, I will be just renaming this as TF and mRNA so that uniformity is there, and renaming the sheets again. So this is TF. So next I will be preparing the third sheet to complete the feed forward loop. Like first is the miRNA mRNA, second is TF mRNA. Now third one will be miRNA TF. So for that I need the input list as TF. But the TF, as you can see, are repeating here. So just I will be fetching the unique ones by going to the Veni, as Veni can help me to get the unique number of TFs. So I can go first to Veni. You can anytime go to the Veni software so that it can list you the unique number of genes. The three TFs were there. So you have to copy this by clicking on the three and selecting all these three TFs. Now we'll be again going to Meerwalk so that the source remains same and there must be uniformity, because for the genes we also went to Meerwalk. So TFs are actually in general only the genes which are actually protein encoding and helping to use to regulate the genes activity. So you have to click on genes again and enter these three TFs, submit and proceed. After that, you will be exporting the CSV file, and I will be taking this CSV file again to the same folder and renaming this as miRNA TF. So after opening this, again, I will do the same steps. So in the binding gap energy, I will be making it in the ascending order and removing all those values or the miRNAs which are having less than 1 as the binding gap energy. And again, checking for the position whether there are any 5 UTR or any other element other than the 3 UTR. So it's safe to go. So I will be taking all these miRNAs and their TFs and saving this in the additional Excel file, new sheet, deleting the between column and just renaming this as miRNA and TF. Now, the one step that has been coming is an important FFL step. is that first we will close this beanwalk file as we don't need this. And we'll be also closing this TF file. So we have created three types of list: miRNA, mRNA, TF, mRNA, and miRNA TF. Now the step that has been left out is that we will be checking that all the miRNAs, these are two columns. miRNA is having one existence in miRNA mRNA and one in the TF miRNA, right? TF, same like TF is having occurrence in miRNA TF, like you can see the here in the second column, and also in the TF mRNA. And likewise, the mRNA or the genes is having one occurrences in TF mRNA and one in miRNA mRNA. So we need to check that the elements miRNA, mRNA, and TF are actually equated or equal, basically same to same in all the three lists. So then only we can create the feed forward loop. So suppose first we take the miRNA, mRNA sheet and copy all these miRNAs and copy and paste that in Veni, clear the older sheets. And so as you can see, the unique ones are 2496. As you can see here, there are 51,921, but it is actually not that because it's repeating internally many times. So the total number is 2496 in case of the miRNA mRNA. But let's check in case of the miRNA TF, how many are there? So in case of miRNA TF, I have copied all the list. It's showing 4,772. It must be less than that. It's 1,001. So actually, as you can see, there is roughly 1 to 1.5 times more miRNAs in the first sheet. So I need to make it equal. So for that, I need this 1,100 miRNAs. So let me clear this. I don't need this one and only the 1,001 I need. So I copy this and I paste it in some new Excel sheet first. Okay, and I will be using some manual operations to equate the number of miRNAs here. So I will be creating an internal extra column and renaming this as miRNA because we are actually, please understand, we are actually lessening the number or making it equal in the miRNA mRNA because these were having more number of miRNAs. So this is the miRNA list I fetched from the, if you remember, the TF-miRNA, and I'm copying this. These are 1,000 miRNAs, and I'm pasting in the additional sheet here, right? You can delete the additional Excel sheet which we created. So you have to click on the first miRNA column, go to conditional formatting, and use a new rule. So your new rule will be use a determine formula to determine which cells to format. So I will be using my own formula, which is equal to, in the small letters you have to type is equal to COUNTIF bracket $B goes to $B and again comma $A1 and close the bracket. And after then click on format, fill, and choose any of the one color. There are many colors here. Click on OK and OK. And you can see this is actually fetched all these miRNAs from the second column, wherever they are present in this first column. But I don't know where they are actually present. So they are actually uniquely present. Like suppose one is in second, third, fourth position, some in six, seven, nine, and like that. So to click on data and click on sort, expand the whole selection. Click on miRNA. In the cell values, you have to change it to cell color. And after that, there is no cell color. You have to click on the drop down menu, click on the green color, and on the top, it's as it is, and click on OK. So it will actually put all the matching miRNAs in the first column on the top. So you can easily fetch that. And we have to do this step repeatedly again and again and again. Then basically all the list elements in all the three sheets have become equal. Once they have become equal, we'll be next creating the feed forward loop. So I will be just copying this, basically taking all these miRNAs and their corresponding mRNAs, or basically the genes. So I will be going down till wherever the color ends, because that have marked all the miRNAs which have matching from the second column in the first column. And it has actually lessened the total number of miRNAs for us. So wherever the color in the green ends, that will be the area that we have to go till. So some miRNAs were left. I'm just copying them because these were left out. So what I will be doing, and as the operation is now completed, so as you can see, there are three columns. So we created an additional column for fetching the miRNAs. So just delete the columns from the between, because we don't need now it. So again, I will recheck the ones so that it's equal or not. So I will again go to Veni first. And what I will be doing, I will be copying the miRNAs first. So this is from the miRNA mRNA. And next I will be copying again from the TF-miRNA. So as you can see, in the TF-miRNAs, these are 1001, but here there are 994. So we can see a difference of around 7 miRNAs here. So I just need to make it equal for the 994 ones. So I will be taking the 994 miRNAs, right? And we'll be doing the operation now in the TF file. So miRNA TF is there. First, let me create an additional column and rename it as miRNAs. And after that, I will be saving all these 994 miRNAs to make it equal from 1001. You use the same formula again. Let's count if $B goes to $B, and after that $A1 is there. So format, and in the fill you can choose any of the color here and click on OK. After that, click on data and you could click sort, expand the selection as it is miRNAs, cell color, and click the cell color here. So as you can see, maximum number of colored here. So these are non-colored. You can remove the non-colored also or take the colored one also. Both are same. We delete the non-colored ones, and after that, we created this additional column. We'll be deleting this. After that, we recheck the miRNA content again. So let's take for miRNA TF. Let's clear the sheet in the Veni first for both the list. So this for the miRNA TF, these are 994. And for the miRNA mRNA, these are 994. So you get a common of 994. Both sides are zero. That means miRNAs are done. Now we'll be checking for the TFs first. So TFs must be equal. So let's go first in the TF gene. So TF gene is having 3. Now I will go to the miRNA TF. This must also have 3. So this is also having 3. That's equal. And there must be some discrepancy in gene because we deleted many number of miRNAs again and again from the miRNA gene. So let me check for the mRNA in the miRNA mRNA. So in the miRNA mRNA, there are total number of 78 genes, right? But if I go to the TF mRNA, there seems to be less number of genes here, which are 14. So I need to actually do what? Do the operation on the 14, these 14 genes in the miRNA mRNA. So first let me copy this and I will going to the miRNA mRNA. Just swap the columns because we need the first column as the ones for which we need to do the operations for. So since we are doing the operations for the mRNA or the gene here, so we have swapped it. So we'll re-swap it back again. Copy these 14 elements here. Now use the same formula again. Choose any of the color and click on OK. And after that, Now it has colored the genes for us, and I will be fetching only the genes. So what we have to do, we have to do again the same steps and fetch the genes for which they have matched with respect to the miRNAs. So copy this and we'll be pasting in another Excel sheet and delete this old one. And after that, since the operation is completed, I will be just deleting this additional between column now. So let me check the genes again. First in the miRNA, mRNA. These are 13. And if we go in the TF MIR mRNA, these are 14. So what one is extra is here in DUFS3. We can delete it from here, right? There are two entries. This will make it equal. Right, so if we again check this by deleting this and re-entering, it is 13 now. So now let me again check for the TFs first. Genes is fine. We are now going for the TFs. So these are 3 in here. Now let me check in the miRNA TF. This must be also 3. And now lastly, we check for the mRNAs. So this is 825 here. This in the TFs. So TFs is 994. So I have to make it equal for the TFs again. So I copy this in additional column here and create the additional column first here. Choose any of the color and click on OK. And after that, click on data and sort the elements. Right. So you have to copy this and delete the additional column in between. And now let me check again for the miRNA in the miRNA TF. A25. And what about in the miRNA mRNA? This must also be A25. So this is equal. Lastly, we again recheck once the miRNA is done. I check for the mRNA, which is the genes. So these are 13 here, and this must be 13. And next, I lastly check for the transcription factors. I copy this from the TF mRNA, and lastly from the miRNA and TF. So once all these are equal, last thing we need to check is like if there is any similarity between the TFs, like both the lists are having the same TFs. So copy the TFs first You can take it from any of the lists. I am taking from MIN and TF. You can take from gene TF also. And the genes which were 88. So there must be not similarity between these two things. So as you can see, 0 is there. So 88 is different, 3 is different. That means we are ready to go for the FFL creation. So what I will do, so I will reswap the columns because first needs to be miRNA and second needs to be mRNA, right? So this is the column which it needs to be assigned like that. Second is TF mRNA. Third is miRNA TF. So we'll be saving this file, save this as an Excel file only. And we'll be just saving in the same folder where we are doing our analysis. So this is FFL. Now you have to go to the Google and download the Cytoscape in your system. The latest version is 3.10.2. You can download this also. I'm using 3.10.1 because there are some additional topology centrality measures which can be seen in 3.10.1 only. So you have to click on the download for actually if you want to download the older versions. As you can see, as you can see, the list is there like old versions. You have to click on old versions, and there are many versions available for the previous ones. I am just taking the one version down, 3.10.1. And for the Windows, there is this 64-bit exe installer, and you can take it for Mac and Unix also. So I have to just open the Cytoscape first after installing and downloading. After downloading, I am just clearing the free unused memory and clear the display. So I have to go on the file option at the top left, go on the import network from the file. And after that, you have to just select the file where you have saved. So this is the FFL file. We take the first sheet, which is miRNA, mRNA. Click on the miRNA. This is the source node. You have to click on the green label to identify it as source node. And mRNA is the target node. Click on the mRNA, and this is a red circular dot, and the name is coming target node. After assigning this, click on OK. So this is the miRNA, mRNA. You can rename the sheet, rename the network here. So this is miRNA, mRNA. Okay, and after that, you have to again go to File, Import, Network from File. Click again on FFL, and this TF mRNA. Second sheet is this. So TF is again the source, and click on the mRNA, that is the target. Click on OK. So TF mRNA name is already there. We don't need to rename it. Third and the final edges, again we go to the FFL. There were three sheets. Third was miRNA as the source and TF as the target. So we go to the network and first rename it as miRNA TF, right? And click on OK. And after that, you have to go on edit, remove the duplicate edges, select all the three networks simultaneously, click on ignore edge direction, click on OK. So it will remove any duplicate edges if there are any from any of the network. Now again, click on edit and remove self loop option. You have to again select all the three networks by clicking control on your keyboard and click on OK. After that, go on edit and view and click on always show graphic details. So As you can see, the first network is having 838 total nodes, as we saw in the presentation, what nodes are, and they are linked by around 1800 edges. Second one is having 16 nodes linked by 16 edges, and likewise 828 linked by 973 edges. You have to go on tools, click on merge and networks, select all the three networks, and we are basically doing the union here. Add these selected to the another box where we have to need to merge and click on merge. So this is the final feed forward loop what we are getting. Always show the graphic details. And this merge network is having 841 nodes linked by the mostly more than double of their linking edges, which are 2800. So now what we have to do, we have to click on the tools and click on analyze network. And basically we need to analyze this network as we need these properties to identify the hub, miRNA genes and TFs in this. So since it's not a directed network, we'll not check or take this, keep it as it is and click on OK. Now you're seeing node table in the bottom, NH table. Okay. So what you need to do, you have to click on the node table and you can see this option export table to file in the left side. You have to just save this in wherever folder you want to save it. I am saving it as FFL network analyzer. So once you have saved it, you have to open that. It's an Excel file. So what we can do, there are three essential centrality measures. First one is degree. So go in the data and sort the degree as per the increasing to decreasing order, or decreasing to increasing order. And as you can see, the RBPJ, first let me open the FFL original list where we prepared that. So RBPJ is the top TF as per the degree, which is just saying that 655 is highest degree. Let's check what's the highest as per the closeness. Centrality is having again highest for the RBPJ. And let's check for the betweenness. These are the three essential centrality measures. So again, you can see RBPJ is the highest element in any of the case. So let me just keep a track and rename it or name it somewhere so that we can just create a marker like RBPJ is the highest centrality measure TF or the key TF or the hub TF based on all the three centrality measures. Now, let me check for all the centrality measures. What's the highest number of miRNA? Okay, so highest number of miRNA, if we sort as per the degree, and if you go as per the degree, it's 67693B. The degree is 9. It's the highest degree as per the all degree elements for the miRNAs. Now let's check for the closeness. For the closeness, we are having nearly equal degree, but all are having nearly same degrees. But let me first check what is the highest number of centrality measures for the gene in our case. So let me first assign the gene list. So second is TBC1D2B. Let me check for as per the degree. So it's a gene. So TBC1D2B is the gene. So it's having 346 as the degree, which is high. And as for the closeness, if we see, again, second highest element, or we can see after TF is the gene only. And as per the betweenness, it's again the TBC1D2B. Let me copy this and we are saving this in one Excel sheet. Now let me cross check whether actually this TF and gene are actually coinciding or not. So RBBJ TBCD12B is perfectly fine. So we can take this, right? So I will be creating one additional Excel file like tbc1d2b and rbpj are connecting as per the first edge list, that is tfmirna. So this is finalized in case of this, and we need to take this, right? But the second and third edge list, which is actually the miRNA TF and miRNA gene, needs to be now finalized. So let's make one file or header like miRNA, and this is TF public, you can take it gene, right? So TF gene we have finalized. TF is RBPJ, right? And the gene was TBC1D2B, but we need to finalize the miRNA now. So let me again go to the network analyzer, and as per the degree, they were showing 6769B3P as per the closeness. 6846 and 6810 is there. So there are variable number of the elements, and the main one is actually the degree. So let me check for 67693B whether the connection exists in the case of FFL or not. So first we'll go to the so the connection exists actually, as you can see, 6769B3P is having a connection here. So we name it here, right? And one thing that has been left is RBPJ. So it's having with RBPG also, as you can see. So we have created one additional subnetwork motif loop. This we actually are fetching for the key miRNAs, mRNAs, and transcription factors from the main FFL. Like as you can see, this is the main FFL, right? But it's hard to interpret like what is actually the key miRNA transcription factors and gene in this. So that's why we have created an finalized hub subnetwork motif loop. Generally, we call that. So we'll saving this in the same folder where we are save the FFL and naming it as FFL loop. Now after that, you have to go on the cytoscape. You can first save this file. So this was actually the FFL. Now again, I'm one by one opening these sheets again from the FFL loop, which I have saved just now. So first is TF as the source node and gene as the target. And again, I will be again importing because miRNA in general is the source node and it targets the TF4 gene. That's why we have saved the time and made only one Excel sheet for that. And after that, I will be merging this. And this is the final subnetwork motif loop in the case. And we can just interpret like in case of the where we took COVID-19 as our data. So out of the all the 88 hub genes where we got RBPJ, which is the transcription factor, is an important transcription factor, the key transcription factor, which is regulating this particular gene that can be a progressive factor in the case of COVID-19 initiation and transcription and further regulation. And somewhere or the other, the MIR 6769B is actually linked or associated to both these key TFs and as well as the genes. So this will be saving as FFL loop. Right. So this is the analysis which we have done today. And we have completed the WGCNA, both modes in the trait and as well as the inclusion of age as the trait. And simply we did the non-trait with nothing inclusive of any of the clinical parameter. And thereby we finalized the final hub genes and as well as moved on to the feed forward loop analysis to fetch the final transcription factors and miRNAs and TFs. So after that, we'll be doing some meta-analysis or playing with some of the multiple data sets tomorrow. And after that, we'll be seeing the RNA-seq in the last session. So if any doubts regarding this session today, please do let me know. Otherwise, we shall be meeting tomorrow.